This is a project that I did prior to going to Purdue. It kind of blended both my doctoral research that was working with organic orchard floor management, and then my postdoctoral research with Steve Jones here in Washington, looking at variety by microbial interactions. So the purpose of this particular study wasn't really to look at improving wheat for wheat's sake, but looking at the value of wheat in an integrated system with apple. So that's my title, looking at varietal selection and then annual versus perennial growth habits on suppression. So soil-borne pathogens are a major cause of yield loss and other problems in agricultural systems. And in apple systems, apple replant disease is a big problem. So it's caused by a complex of several different pathogens and parasites, and it negatively, these pathogens build up over the life of an orchard. And they generally don't impact mature trees, but when you try to remove those mature trees and plant new young trees in response to maybe the age of the tree, or you want to put in a new variety, the young trees are really negatively impacted. They have poor establishment, and it negatively impacts the productivity of the orchard throughout the life of that system. And so because it's such a big problem, orchardists, even organic orchardists, will remove their land from production and use pre-plant fumigants that are these very toxic chemicals that are harmful to the environment and workers, but they also completely sterilize the soil, so killing all the pathogens as well as the beneficials. So we need some alternatives to that problem. So numerous studies have shown that if you rotate your crops, obviously with cover crops or mixed hay species, you can reduce soil-borne pathogens in the system. And we've seen in some of these studies that the suppression due to these cover crops is correlated with changes in the general activity of the microbial community, as well as the composition of that community, including members that have been well characterized as biocontrol agents, like these fluorescent pseudomonad species and bacillus and mycorrhiza. And these help suppress plant disease through a number of mechanisms. For one, competing with pathogens for nutrients, or even producing toxic compounds that are antagonistic to some of the pathogens. So plants have very intimate associations with microbial species in the soil. They actually take a lot of the material that they get through photosynthesis and actively release that through their roots as exudates. And these are chemical signaling compounds where they can call in the microbes, or actually food sources like carbohydrates to support these. And it varies a lot by plants, but it's estimated that up to 30 plants will release up to 30% of their food sources to signal these bacteria in the soil and fungi. And the amount and composition varies both by species as well as genotype. And a recent study by Gaudry et al. Actually found that even a single gene mutation in the exporter out of that root structure that's releasing this compounds can dramatically change the whole microbial community that's associated with that plant. So soil-borne disease suppression using cover crops for that obviously depends on plant species. And there's a number of species you can use in this study. Sorry, the light doesn't seem to be working very well. But you can see up near the top is the control, where they had the soil-borne pathogens, and then they tried various cover crops to reduce those pathogens. And you can see wheat is actually one of the better ones in helping to suppress soil-borne pathogens, in this case with snap beans.
So were the ones at the bottom were the most effective? I didn't quite understand.
Yeah.
To go over that.
Yeah, so try to go back a little bit. So this is top, so the root rot severity. Down here at the bottom, number 1 would be no root rot severity, and 9 at the top would being the most. So as you go down this list, like rapeseed is the best in this case, and then wheat was actually pretty good as compared to some of the other ones that are more standardly used, like ryegrass.
But like what, for instance, most of my soil-borne pathogens are in brassica crops. Rapeseed would be a poor choice for a cover crop if I'm trying to break disease cycles.
Right. And
go with one of those grains in there?
Yeah, and it depends. You know, there's different pathogens attack different crops. And so some are very broad hosts. They can attract a lot, whereas with your brassicas, it might be specific species. So yeah, you would probably want to use a brassica in that case, and you might want to use a wheat or a grain crop to help disrupt the pathogens. Yeah.
It seems like we were just talking about how white clover increased the ability of soil-borne pathogens in the soil. I was wondering if there's a theory of why that is.
I don't know exactly. I know, they have a lot of available nitrogen, a lot of the saprophytes, and, when you have a lot of nitrogen in the system, some of those species can grow really rapidly. So, yeah, I don't know the exact answer to that question.
Why do white clover and alfalfa be so different?
Well, I think, in terms of the way I've looked at it, it's because they're supporting a different host within the community. So maybe alfalfa has something in it that can be better than a white clover.
Just repeat the question.
Oh, I apologize, yes. So the question was, why would a white clover not be as suppressive as an alfalfa, for example? Or why would white clover be not disease suppressive compared to some other ones?
Is the wheat and the rye grain winter wheat and winter rye?
In this study, they didn't actually specify that. They probably do in the paper. I didn't look at it. In my research that I'm about to talk about, I looked at a range of winter wheat and spring wheat varieties. Okay.
You probably already addressed this maybe, but I'm just curious about how much of this ranking could be correlated to just pure biomass in the soil, and how much X amount of organic matter incorporated in the soil will could suppress some of these pathogens.
Yeah, I think there's definitely lots of benefits to building soil organic matter and health, and perhaps correlated with changes in the microbial community. So when you compare, say, a conventional system and organic system, as you bring that in, you're going to have food source for the beneficials. In this instance, we're actually looking at differences between the species. So we know from, I guess, the previous slide, there's a lot of differences in how you would support that community. So we're trying to correlate whether these plants are specifically targeting these specific microbial species versus that. But I think there's definitely benefit in bringing more organic matter into the system and helping suppress disease. Okay. So back to this study, we're looking at breeding. So can we use breeding to enhance, make a better cover crop? So we know that wheat can help to suppress the pathogens in the soil. And there's also been some studies that have shown that wheat varieties differ in their composition of the root exudates and then the amount of these beneficial biocontrol agents. So in this case, the fluorescent pseudomonads that produce this toxic compound 2,4-DAPG, which is antagonistic to pathogens. And Gu and Mazzola actually screened 13 different wheat varieties and found differences in how the varieties support these, and they were correlated with suppression of apple replant disease in greenhouse studies, but it didn't work as well in the field.
Sorry, yeah. Question. So the screening was mostly done in greenhouse, kind of, and then it didn't translate to the field. Like, how robust was that variety?
Yeah, well, the reason that they thought at the time that it didn't work in the field was, as I'll show, that annual weeds have very shallow root systems, and so this is why we started looking at perennial wheat, because they have more deeper root systems. So if you have using a cover crop that has a shallow root system, are you really transforming the microbial community as well as you could? Or when you're in a pot system, obviously you can fill that pretty well with an annual wheat. So yeah, hence why we thought perennials might be a good idea. Okay, so we wanted to know then, do breeding conditions, the select conditions under which varieties are bred, have an impact on their ability to interact with microbial species. So I went to the literature and found some really interesting studies, I thought. There was one by Toby Kears who looked at breeding soybeans. So she looked at soybean varieties that had been bred prior to the use of high input fertilizers and things, fertilizer organics, or breeding programs, and then ones that have been bred more recently under high nitrogen conditions. conditions. And so these soybeans associated with rhizobia, when you have a lot of nitrogen in the system, the plant will not form those associations because it's getting the nitrogen elsewhere. And so theoretically, perhaps when you're selecting, you might select varieties that aren't better able to form those associations. And what she found was that these modern varieties could no longer tell the difference between rhizobial symbionts that fixed nitrogen, were very efficient in fixing a lot of nitrogen, and ones that weren't. And so that, whereas the older varieties did know how to discriminate and find these microbes in the soil that produced a lot of nitrogen. Alternatively, Baldini, who was selecting for sugarcane varieties under very low input conditions in South America, so when there wasn't a lot of nitrogen, and selecting for these high yielding varieties, found that they had actually inadvertently selected for varieties that could form associations with diazotroph and sort of free-living nitrogen fixers and fix a lot of nitrogen. So we thought perhaps maybe there'll be some difference in the wheat varieties that we look at. So as Sieg was talking about earlier, one of the reasons we think in the previous studies that perhaps when you took it out into the field you couldn't really modify the microbial community as well is that annual wheat has very shallow root systems. So we might not really be exploring that profile and affecting change. Whereas perennial wheat, which is a cross between annual wheat and its perennial wheat ancestors, has these much deeper and larger, more extensive root systems. And also, unlike annual wheat tends to be susceptible to some of these pathogens that we were looking at, the Rhizoctonia and Pythium. Whereas in the perennial wheat, we found that they're actually tolerant to some of these pathogens. So perhaps perennial wheat might be a better way to start to control pathogens in soils. And it also has other benefits in that it regrows after cutting and environmental benefits that I'll direct to Sig later to talk about if you're interested. Yeah?
It regrows after cutting. Is there root shedding after cutting?
But we have so many rhizotones, like little tubes, and look at that right now. We're going to students at Michigan State. Maybe next year.
Okay, so moving on. So the goal of this study was just to determine whether selection conditions do impact how we're selecting wheat. As we select wheat varieties in different systems, are we affecting their interaction with beneficial microbial communities that might help suppress disease? And then what does that do for apple seedling health? And so we collected soil from orchards here in Washington State that were infested with these pathogens, and we did some greenhouse trials. We had the control pathogen. We used an annual ryegrass that tends to be used a lot for disease suppression. And we selected 4 random annual historic wheat varieties. So these were varieties that were selected prior to the widespread use of agrochemical fertilizers and pesticides that are being used in the organic breeding program. And then we had 4 annual high input wheat genotypes. So these were varieties that had been selected recently under very high input conditions. And they had been identified in Mazzola's previous research as to having a greater ability to enhance populations of these beneficial antagonists. Then we had 4 varieties from the advanced organic breeding program that are selected under much lower input conditions, more stressful environments. We had a wheat relative and then one of 4 perennial genotypes from the advanced perennial breeding program. And so we grew these, we did a couple cycles in the pots, and after that, after the wheat was done, we planted apples into those systems, looked at apple health after 10 weeks, and then took one group from each of those and looked at the microbial community that was living in the rhizosphere and repeated the experiment. We also did field studies at 2 sites in Washington State, one near Wenatchee and one near Pullman, Washington. It had documented infestation of pathogens. And with our greenhouse, well, when we brought it into the lab, we either pasteurized there or fumigated in the field with some brassica seed meal, seed meal as a fumigant. And we had that high input annual wheat genotype. Yeah.
Could you explain what's pasteurized?
Yeah, so pasteurized is just a process where you take steam at a high temperature through the soil to try to kill everything that's living in that system. So it's kind of a check that we can say there's not anything. And this would be similar to what growers are doing in the field when they use chemical fumigants. So they're just trying to kill every living organism in that soil. So this is not something we would normally do in the greenhouse, but as purposes for our study so we can really see what's happening. Okay, so we had the high input annual wheat genotype Pinawawa that was previously identified as being very good at enhancing these microbes. We had the wheat ancestor, T. intermedium, and then we had to use a mixture of perennial wheat genotypes, so this is still a breeding program and we don't have large amounts of seed to use in field studies. And then we couldn't include the organic because also this was a breeding program and we didn't have a lot of seed to use in our field studies. So this is the results of what we found in our greenhouse studies. These are all the different varieties. We've got the control, the pasteurized. So this is apple seedling health after the wheat was grown in there. So with the pasteurized, where we effectively killed all the pathogens in that system, you can see it did quite well. And then we had all of these different wheat genotypes. And there's a lot of variability between the genotypes and what their impact on that apple seedling health. And we started to see some trends with the historic varieties. Really didn't do a whole lot to enhance those beneficials. The high inputs did a little better, particularly that Pinawawa. And then our organic ones started to do even better. We had one there, the Madison Onus, that did quite well. We thought we probably depleted the nutrients quite a bit in this study, so had we added a little more fertilizer, we might have seen some even better impacts to the apple seedling health. So this is just a graph then showing the different, when we pulled all the varieties together and averaged how the historic varieties did in improving apple seedling health against the high input ones, and then the organic ones, you can start to see a trend. And this, I want to point out that these high input ones were ones we already knew were pretty good at enhancing these populations in the soil, whereas our organic ones and historics were more random. So it's a small population size, but I think we see an interesting trend. This is our perennial genotypes, how they came out. And we had a lot of variability within the perennial genotypes, so they're still segregating. So some pots were having a large effect, some pots weren't, and this graph doesn't really show that very well. But we did have one variety, P006, that performed really well at boosting apple seedling health, even as good as the pasteurized treatment. And when we looked at then changes, yeah.
I'm just curious about what the physical benefit was for you and the wheat in the pots, and then what you do and how was that actually going to happen in the field situation?
Well, we were planting the wheat in the pots just like you might plant wheat as a cover crop in the field soil. And so trying to affect those changes, and then we took the wheat out, we cut it, stirred it up like tilling, and then planted the apple in there just like you would do in the soil. The advantage of the greenhouse test is that we can control things a little bit better, variables that, soil type, we can make it more uniform. We're not worried about a tornado coming through or other variables like that. So.
You guys did the mostly the soil.
Yeah, so for the annuals, we cut it right at the surface and then mixed it all in. So we never grew it to, the point of the study was not to produce wheat, so it was just like you would do a green manure in the field. With the perennial, we cut it and took the biomass off, like you might look at that for pasture, and then it was allowed to regrow back. The other annuals, we would plant it again just to get some cycles in there. So yeah, we saw some differences with the perennials. And then we looked at changes in the microbial community, so what was happening as a result of these things. And we found that these top varieties did do very well at increasing the fluorescent Pseudomonas populations and then decreasing our pathogens. So we saw a correlation with the improvement in our apple seedling health with the modification in the microbial community. So in the field situation, our Penawawa actually did really well. It was a spring wheat. It established well, grew well, and we actually grew that through the sickle. And it was very good at suppressing the Pratolynchus penetrans, which was the biggest problem at our Fuller site. Fortunately, our perennial wheat, oh yeah, go ahead.
Here's one question online. Somebody asking if you think that the nitrogen content on Sinapis alba against the root biomass, and also wondering whether perennial wheat is resistant to
Oh, I can't, okay, is perennial wheat resistant to G99? I don't know the answer to that. Say, do you have any idea?
Perennial wheat is a whole new type of crop, so there's a whole range of resistance that we're still learning about.
Yeah, in response to the other question about the seed meal providing nitrogen, absolutely. Those seed meals have about 7 to 8% nitrogen. And so Dr. Mazzola is doing a lot of work right now looking at these seed meals, and I've done some work looking at them as biological weed suppression. And so you do get the benefit both of this nitrogen fertilizer as well as helping to suppress pathogens and weeds. So yeah, if you can find it, it's a good compound to use. But it can also hurt your target crop. So you have to be careful when you use it. You know, you need to put it on, just like when you put in a cover crop, you need to wait a little while for that cycle to go through before you come in and plant like your broccoli or some other crop, because it could have a negative impact. Okay, I wanted to show this real quick. So the perennial, unfortunately, we had to plant it in the spring rather than the fall. Like we normally would like to plant in the fall, let it grow through the winter and really establish itself in the spring. But we had to plant it because of logistics with our experiment in the spring. So we really got pretty poor establishment. But despite that, we saw really dramatic decreases in the Pratolynchus penetrans populations, and even when that was segregating. So we were pretty excited about that. So in conclusion, I think wheat is a valuable crop to use in rotation to help suppress disease. You're going to get some benefits, I think, out of it, and hopefully suppressing some pathogens if that's an issue for you, as well as it's a great crop. And while this study was done in apples, a lot of these pathogens do affect multiple crop species, so it will have value. I think breeding programs can be designed to exploit beneficial microbial interactions. It hasn't been something that most breeding programs, I think, have looked at, but we're seeing some really great results out of different studies showing how varieties are very different in how they associate with the microbes in this case, and how they increase these antagonistic species that both help wheat as well as other subsequent crop reduce disease pressure, and then with nutrients too. So there's been some studies to show wheat varieties differ in how they associate with mycorrhiza, and I've actually done some work also looking at wheat varieties and how they associate with azospirillum, which is a bacteria that helps the plant to fix nitrogen. So I think we can start to target this. And then I think selection under organic conditions is a good idea. There's been a variety of studies who have shown that the microbial community is much more diverse, active under organic systems as compared to conventional. And we know that soil microbes in controlled studies can influence plants' phenotypic traits, so their yields, their disease resistance. And we know also that the varieties that perform best in organic systems are not always the ones that perform best in conventional. So perhaps the reason for this is because we have these more robust communities in organic systems, and we might be selecting varieties that based on some of the traits we're seeing because of these more robust communities. So this is something that hasn't been very well tested, but we're hoping to, we will be working on that in the next couple of years and hope to have some better answers for you. And I also think the continued development of perennial wheat is a good idea both to help reduce disease as well as to bring broad agronomic and environmental benefits to the system, and I would point you to SIG to talk about that. I want to thank the Organic Farming Research Foundation for supporting this research, and my collaborators, Dr. Murphy, Dr. Mazzola, and Steve Jones, Dr. Jones. Okay.